طلاب الاونلاين سامعيني؟
دكتور
سامعين شايفين السلايدات؟ شكرا لك. بنات رجاء القهوة والهاي ممنوع تدخل القاعة، ممنوع. انتم شايفين كيف؟ السلام عليكم. أنا الدكتور جهاد شنيدات، ما شفتوني قبل مرة. كنت أدرس أونلاين. بالنسبة للبايوكيمستري للجي آي تي قسمناهم بأربع محاضرات، أنا راح أعطيكم الأربع محاضرات على نفس النهج اللي حكيت لكم إياه بالسنة الأولى. إذا متذكرين بالسنة الأولى حكيت لكم أنا يعني من السنة الأولى للسنة الثالثة عندي نفس الشيء. بالنسبة للجي آي تي اسكت يا بني. بالنسبة للجي آي تي عندكم أربع محاضرات. المطلوب منك السلايدات فقط، وإذا أعطيتك نوت النوت، يعني المطلوب يعني لا تقرأ external نوتس أو بوكس أو تسمع الريكورد. أي معلومة بقولها مش موجودة بالنوتس أو السلايدات مش مطلوبة. واضح؟ هذا يعني لا تيجي تسألني بعدين الشغلة الثانية. كل شيء موجود بالسلايدات أو بالنوت مطلوب، إلا إذا أنا حكيت لك إنه هذا مش مطلوب. يعني الاستراكشرز، الفاصلة، النقطة بدك تكون عارف، لأنه أنا ما بطلب منك لا ريكورد ولا بطلب منك يعني ترجع لمراجع خارجية ولا شيء ولا أي شيء. فقط دائما وأبدا إما بتقرأ عن النوتس وبتراجع بالسلايدات. بالنسبة للجي آي تي ما في نوتس، ما فيش نوتس، سلايدات فقط. فبتمسك السلايدات بتفهمهم بتحفظهم بتسوي بيهم اللي بدك اياه. لا تسالني كيف شو هاي مطلوبه ولا مش مطلوبه، كل شيء مطلوب لانه السلايدات فقط. واذا جمعت السلايدات كنوت والله الاربع محاضرات بجوز ما يجي نفسها كم من صفحه. واضح؟ فمش راح يجيك اي شيء ان شاء الله خارج السلايدات، لازم. واضح؟ اوكي. مثل ما حكيت لكم البايوكيمستري بالجي اي تي في اربع محاضرات انا راح اعطيكم اياهم. المحاضره الاولى والثانيه عن الدايجيشن، ده كاربوهيدراتس، بروتينز وفاتس. كلير؟ المحاضرات الثلاث واربعه راح نحكي عنهم في وقتها. اوكي. والله يا بنات الأخير رجاء أنا عندي يعني سيستم واضح، جزء أنا ما درستكم هذا ولا نفس بالمحاضرة بتاتا، رجاء واضح؟ Okay. There are 7 types of nutrients. So in nature, there are 7 types of nutrients, which include carbohydrates, proteins, fats, vitamins, minerals, water, and fibers. In today's lecture, inshallah, and tomorrow's lecture, we will study carbohydrates, proteins, and fats. لغينا محاضرة لأنه أخذت الفيتامينز، نوع من الفيتامينز بالسنة الأولى. وبالنسبة وأخذته عن المينرالز والأمور هاي، فلذلك كان في 5 محاضرات من الجي اي تي لغينا. صفوا 4 محاضرات إن شاء الله. So in today's lecture and tomorrow's lecture, inshallah, we will study carbohydrates, proteins, and fats. We will take a slight short notes about their structure and their digestion. Okay. At the start, we will study the food guide pyramid. Food guide pyramid. What do we mean by food guide pyramid? From the name, food guide, it guides us to how much we should eat of each of every ingredients of food every day. As you can see, this is the pyramid, the base of the pyramid, the next level, and the top. The base of the pyramid include bread, cereal, rice, pasta, fruit. How much we should have every day of these things? 6 to 11 serving. When we say bread, cereal, rice, pasta, and so on, what do we mean by them? They belong to which ingredient of food? Carbohydrates. So we should have, in general, of these foods, 6 to 11 servings. When we say servings, what do we mean by a serving? Serving is different between one ingredient and another. To your left hand of the slides, I give some examples of the serving. For example, when we say grain, حبوب, grains, one slice of bread, شريفنا العربي, slice, one slice is 1 serving. Half cup of cooked rice is 1 serving. Half cup of cooked veg is 1 serving. 60 grams of cheese or 1 cup of milk is 1 serving. 60 to 80 grams of beans or meat is 1 serving. This is the serving. So serving is different between one ingredient and another. So the base of the pyramid, it includes generally the carbohydrates in general, bread, cereal, rice, pasta, and so on. How much we should have? 6 to 11 serving. We will go to second level. Second level, it includes 2 types of the food, fruits and vegetables. Fruits, we should have 2 to 4 servings, while veg, 3 to 5 servings. The third level, it includes meat, poultry, fish, and milk, yogurt, cheese, which we call it the dairy product. Meat, poultry, fish, and so on, 2 to 3 servings, while the dairy product, منتجات الحليب, dairy product, 2 to 3 servings, same as the meat. The top of the pyramid, it includes fats, oil, and serving. Use sparingly means as less as possible. So, as you can see, the base, we should have more. As we go up, less, and the least is the top of the pyramid. We should have as much less as possible. As much less as possible because it includes the fat, oil, and sweets. Clear? This is the food guide pyramid. It tells us how much servings, how many servings of each of ingredients of the food we should have daily. Clear? Now, as we said, we will study carbohydrates, proteins, and fats. In today's lecture, we will start with the carbohydrates. Now, what is the function of carbohydrates? Carbohydrates has many functions in our body, which include energy. The sugars are energy, glucose. Fibers. See from now, undigested polysaccharides, they form the fibers. So what is the fibers? Digested polysaccharides. The undigested polysaccharides, they form the fiber, and we will take short notes about fibers. We will study some of them. Then recognition and adhesion. They form parts of the receptors. This is why recognition. They are parts of the cell receptors on the cell membrane. And adhesion between cells as adhesive substance between the cells. This is why carbohydrates with a protein attached to a protein, we call it glycoprotein. So what is glycoprotein? Carbohydrates with a protein. So glyco always means carbohydrate. Glycoprotein or glycolipids, carbohydrates attached to lipids here and protein here. Now, this is just an introduction. The major source of carbohydrate comes from plants, not animals. So animal food, the animal source means the meat does not contain that much of carbohydrates. So from our food, from where mainly we get the carbohydrates? From plants. The major source of carbohydrates from animal is the lactose sugar, which is the milk sugar. So in our food, from where we get normally the carbohydrates? From the plants. Food or source. Okay, the animal has some carbohydrates? Yes. Example is lactose, the milk sugar. Also, the glycogen in the liver of the animals and some in the muscles. Oxidation. This is just a general introduction to carbohydrates. 1 gram of carbohydrates, once it is oxidized, it will give 4 kilocalories of energy. While the protein also gives the same energy. See, the fat will give more than double, 9 kilocalories per gram. This is why the major source of storage of energy in our body is the fat, not the glycogen in our liver, not the glycogen. Glycogen is made up of what? Which is mainly which is the glucose. So glucose, when hundreds, thousands of glucose come together in our liver, they form the glycogen. So the building block of glycogen is glucose. So the major storage form of energy in our body is the fat, not the glycogen. Not the glycogen. See how much the fat store energy. 1 gram gives 9 kilocalories, while 1 gram of proteins or carbohydrates gives only 4 kilocalories, less than half of that of the fat. Now, carbohydrates, as you studied in the first year, is classified into monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides are the simplest sugars. So monosaccharides are the simplest sugar. They can't be broken down to a simpler one. And they are the building block for oligosaccharides and polysaccharides. So what is the building block for oligosaccharides? Monosaccharides. For polysaccharides, monosaccharides. Exactly as the amino acids in proteins. Exactly as the amino acids in proteins or enzymes in general. Clear? When 2 to 10 monosaccharides they are attached together, they will form oligosaccharides. When more than that, they will form polysaccharides. The most important polysaccharides in our food as a source of energy and sugar is glycogen in animals. The glycogen of the plants, we call it starch. Starch. So in animals it is glycogen, in plants it is starch. This is the matter. So this is the monosaccharide, which is the building block for oligosaccharides and polysaccharides. As we said, this is all our introduction only. يعني لحد الآن ما دخلنا هي introduction انتوا أخذتوها بالسنة الأولى ولكن لأنها بتشكل بيس اللي راح ندرسه. Now, we will study some disaccharides. We will take 6 examples of disaccharides. 6 examples. Maltose, cellulobiose, isomaltose, trehalose, lactose, and sucrose. 6 examples. Clear? Now, see here. لاحظوا معايا ملاحظة دقيقة. Maltose, cellulobiose, isomaltose, trehalose. What is the building block of them? On the glucose. So, maltose is made up of 2 glucose. When we say disaccharides, it means 2 monosaccharides attached to each other. This is the disaccharides. Trisaccharide, 3, tetra, beta, and so on. So, disaccharides, it means that the sugars that are made up of 2 monosaccharides. So, every one of these 6 examples are made up of 2 monosaccharides. What I want you to make sure and concentrate on is that the first 4 disaccharides, they all are made up of 2 units of glucose. What is the difference between them? It's the bond between them. So maltose is made up of 2 glucose. Cellulobiose is made up of 2 glucose. Isomaltose is made up of 2 glucose. Trehalose is made up of 2 glucose. So all of them, the first 4, are made up of 2 glucose. So what is the difference between them? Is the type of the bond between them. What is the bond between maltose in maltose? Alpha 1,4. And we will see what that means. In cellulobiose, beta 1,4. In isomaltose, alpha 1,6. In trehalose, it is alpha 1,1. It is alpha 1,1. So although the first 4 disaccharides are made up of 2 glucose units, the difference between them is the type of bond between the glucose units. The 5th disaccharide is the lactose, which is glucose galactose, the milk sugar. What type of bond? Beta 1,4. Sucrose, composed of glucose, fructose, alpha 1,2. Now, why we study the type of the bond? Why the bond here is important? Why? Like in GIT, digestion, see. Digestion, what do we mean by digestion? Breaking down the bond. So what is the digestion of carbohydrates? Breaking down this bond. Every bond has its own a specific enzyme. So there are some enzymes that are specific for alpha-1,4 glucose. See, we have beta-1,4 has different type of enzymes. Alpha-1,6, alpha-1,1. See, beta-1,4 but glucose galactose has different type of enzyme. So what is the digestion? When we say digestion, breaking down the bond between monosaccharides. This is the digestion. And before we enter into the digestion, this slide gives you the glycosidic bond. What type of bond between the monosaccharides? Glycosidic bond. Glycosidic bond is a covalent bond. As I told you from the first year, what is the peptide bond? It is a covalent bond. What is the glycosidic bond? It's a covalent bond. What is the phosphodiester bond in DNA? It's a covalent bond. But to differentiate between them, the scientists gave them different things. So when I tell you peptide bond, there is no need to explain more, because peptide bond is the covalent bond between amino acids. Phosphodiester bond, it is a covalent bond between phosphate and sugar. There is nothing to say more. Glycosidic bond between two monosaccharides. So scientists give different covalent bonds, different names, so for you to understand directly. When we say peptide bond, it is the covalent bond between two amino acids. Glycosidic bond between two sugars in general. Now, for glycosidic bond, see, this is important. ركز معي. Monosaccharides مثل ما درسته انتم بالسنة الأولى. Monosaccharides are divided into two groups, aldoses and ketoses. Aldoses, CHO, ketoses, C double bond O. So the monosaccharides are divided into two groups, aldoses and ketoses. Aldoses such as glucose, ketoses such as fructose. All of them are 6C6H12O6. What is the difference between them? In aldoses, the functional group is aldehyde, CHO, which is normally present on carbon atom number 1, always. While in ketoses, ketone functional group C double bond O is always present in carbon atom number 2. The carbon atom that has the functional group, we call it anomeric carbon atom. So what is the anomeric carbon atom? The carbon atom that is bonded with it, the functional group. It is always in aldoses, carbon atom number 1, always. And in ketoses, number 2, always. For glycosidic bond, see why we are talking in the base. Always the glycosidic bond is between the anomeric carbon atom of one sugar and any other atom of the next sugar. So the glycosidic bond must involve what? Anomeric carbon atom, a minimum of 1. Clear? So see here. We are talking about glycosidic bond in maltose. Maltose is made up of glucose. See here. The anomeric carbon atom is carbon atom number 1. This is the carbon atom number 1. And this is the carbon atom number 1 in the next sugar. The one in the pink color is the anomeric carbon atom. In aldoses, see, it is between carbon atom number 1 and carbon atom number 4 of the next sugar. This is why we call it alpha-1,4. Alpha because the OH is down the ring, or the bond is down the ring. If it is up, beta. So why we call it alpha? Because the bond, as you can see, is down the ring. Clear?
Why we call it alpha 1,4? Because the bond is between carbon atom number 1 of this sugar and 4 of the next sugar. Clear? This is the glycosidic bond. Can the glycosidic bond between carbon atom number 3 and 4? Why? Right. It must involve the anomalic carbon atom. This is a must for glycosidic bond. It must involve what? Anomalic carbon atom. And aldose is always the carbon atom. Number 1 is the anomalic carbon atom. While in ketosis, carbon atom number 2. This is why, as you can see here, this is the chemical structure of magnesium. This is the chemical structure of disaccharides. See, the first one in lactose. Does the bond down the ring or above the ring? Above, so it is beta. It is aldose, so it must involve 1. And for glucose, it is lactose. See maltose. Glucose, alpha 1,4, the one that we studied, and so on. This is isomaltose, alpha 1,6. See? يعني لو بيجيكوا سؤال بيجيكوا على نفس الشيء بس انك تميز بيناتهم. Which is which? See here. See sucrose. Sucrose is between glucose 1 and fructose number 2. Fructose is it aldoses or ketosis? Ketosis. So its anomalic carbon atom is number 2. A very important note here, ركز معي هنا. See here. Let us talk about lactose. Lactose is between carbon atom number 1 and 4 of the next sugar. So the anomalic carbon atom of the next sugar, is it free or not? Is it bonded with anything? No. When the anomalic carbon is not bonded with any other sugar, we call it a reducing sugar. So this is a reducing sugar. So what is the reducing sugar? The sugar with a free anomalic carbon atom. A free means unbonded. C maltose, alpha 1,4. C sucrose. Is it reducing or non-reducing? It is non-reducing sugar. The only 2 non-reducing sugar, we have 3 halos, it is alpha 1,1 glucose, and the sucrose, glucose, fructose. This is the only non-reducing disaccharides. While the rest are reducing, because all of them, the green, are the free anomalic carbon unbonded. Clear? Now we will start with the digestion. Now we will start with the digestion. See here. Polysaccharides. The most important polysaccharide in our food is the starch. Starch is made up of glucose only. Glucose only. See the next slide. What type of bond between the glucose? We have 2 types of ingredients in starch. See here. Polysaccharide is made up of amylose, 20%, and 80% amyloprotein. But all of them are glucose only. What type of bond in amylose? Alpha 1,4 glucose, alpha 1,4 glucose, always. So it is a straight chain, unbranched. While amyloprotein is the branched one. Clear? See here. What type of bond in amylose? Alpha 1,4 glucose. What type of bond in amyloprotein? Alpha 1,4 and alpha 1,6. Wherever there is a branching point, it is alpha 1,6. A very important note here, see.
alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4. Only the branching point is alpha 1,6. Only the branching point, this one is the alpha 1,6. So this is the alpha 6, alpha 1,6, while this is alpha 1,4, alpha 1,4, alpha 1,4, alpha 1,4. Clear? So the starch is made up of amylose, alpha 1,4, glucose, 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 and so on, and amylopectin. What type of bond? Alpha 1,4, and the branching point only alpha 1,6. While the starch always is made up of only glucose, only glucose. Now, digestion. Glycogen has the same structure as of the starch. Digestion. In general, we call the enzymes that digest the carbohydrates glycosidases because they break down glycosidic bond. So in general, we call them glycosidases. From the start, see here, cellulose or cellulose, the one that in disaccharides. Glucose beta 1,4. It is always in cellulose or cellulose. Our body does not have the enzyme to digest them. Why? So our body can't digest cellulose. Why? Because our body does not have the enzymes to break the bond between glucose beta 1,4, glycosidic bond. Clear? So we can't digest this one. Now, digestion. We will start with the digestion of carbohydrates. Digestion of the carbohydrates starts in our mouth through salivary alpha amylase. We have pancreatic alpha amylase also. So we have the alpha amylase is produced by salivary glands in our mouth and by the pancreas, as we will see. And both have the same function and structure. Clear? See here. Alpha amylase, what you need to know about. Alpha amylase is endoglycosidases. What does this mean? See here. This is a polysaccharide. On the top, this is the polysaccharide. If the enzyme start to break down the monosaccharides from the last sugar monosaccharide, we call it exoglycosidases. But if the enzyme start to break down within the molecule, we call it endoglycosidases. Clear? So exo, it means it starts from the last monosaccharides, from the last sugar. If it starts from the within the molecule, not the last, we call it endoglycosidases. So salivary alpha amylase is endoglycosidases. It is specific for alpha 1,4 glucose. So it can't break down any other bond. Only alpha 1,4 between two glucose. And it functions best at pH nearly 6.7. And it needs chloride. This enzyme needs chloride for its activity. Clear? So, alpha amylase, endoglycosidases, it breaks down alpha 1,4 glucose. It needs chloride and function at which we add 6.7. See here. Normally alpha amylase break down the starch into disaccharides or trisaccharides, or which we call it alpha dextrin, short chain of monosaccharides. So alpha amylase, salivary alpha amylase,
break down the starch, the polysaccharide, into what? Into disaccharides, trisaccharides, or short chain of monosaccharides, or short chain of monosaccharides. Clear? This is its function. It is specific for which bond? Alpha 1 for glucose. Now, in the stomach, there is no digestion of carbohydrate. Only some hydrolysis to the sucrose. Otherwise, there is no digestion due to high acidity. The high acidity break down the salivary alpha amylase. So no enzymatic digestion for carbohydrate in our stomach. No enzymatic digestion. In the small intestine, there is the pancreas secretion, and we have also disaccharidases produced by the brush border of the small intestine. So the carbohydrates start, the digestion of the carbohydrates start where? In the mouth. Stop in the stomach. Then continue in the small intestine by pancreatic secretion. Pancreatic secretion, and this is general for all digestion, whether carbohydrate, protein, or fats. The bicarbonate has same function. What is the function of bicarbonate in digestion? Increasing the pH. Always this is the function. Because the food that comes from our stomach is highly acidic. And the pancreas enzymes need a higher pH. Who increases the pH? The bicarbonate. This is a very important rule. And this is always the rule and the function of bicarbonate. Increasing the pH of the food that comes from the stomach. Thus, creating a proper environment for the function and proper pH for the function of the pancreatic enzymes and the rest of the enzymes. Then, as we said, pancreatic alpha amylase. So, regarding the digestion of carbohydrates, pancreas has two secretions. Bicarbonate increasing the pH, and alpha amylase, same as the salivary alpha amylase. Nothing to say more. It is the same. Disaccharides of the brush border. So the brush border this is the brush border of the small intestine to increase the surface. It is made up, there are thousands of villi or villi, British and American. Villi, thousands of villi. And every villi has thousands of microvilli on it. This is present in the small intestine. The epithelial cells of the small intestine produces enzymes. In general, it is known as disaccharidases. We will take few examples about 4 enzymes that is produced by the brush border. This is area, we call it in general, villi and microvilli in the small intestine. We call it the brush border. Yeah. Okay. This is just an introduction only. So, disaccharides of the brush border of the small intestine. As we said, the epithelial cells produces disaccharidases. The disaccharidases in general are specific for disaccharides and trisaccharides, or maybe a little bit more monosaccharides. But they are very specific to disaccharides. The first disaccharidases is glucoamylase. Glucoamylase has two functional groups, two catalytic sites. This is what you need to know. The second thing, Glucoamylase is exoglycosidase. It means what? From the large sugar start degrading the polysaccharide. See here also, it always starts from the non-reducing end. As we said, see, suppose that we have maltotriose. It is maltose with one extra monosaccharides, 3 monosaccharides, trisaccharides, 1, 2, 3. 1, 2, 3 of glucose. What type of bond? Alpha 1, 4. This is what we are going to say now. As you can see, maltotriose has 2 ends, one reducing end, one non-reducing end. What do we mean by reducing end? Free anomeric carbon atom. And the end that does not have a free anomeric carbon atom, we call it non-reducing end. So a reducing end means that the sugar here, which is always beside the oxygen, because it is carbon atom number 1 in aldoses and 2 in ketosis, and it is always between, very close to the oxygen. So this is the anomeric carbon atom, and this is the reducing sugar. While the other end, see, is far away from oxygen. So it is the non-reducing end. Always glucoamylase start from the non-reducing end. So if there is, see, 2 glycosidic bond, it will start always from the one close to the non-reducing end. So it will break down this glycosidic bond, number 1, then number 2. Clear? So, what you need to know about glucoamylase. It has 2 catalytic sites, 2 active sites. It is exoglycosidase. It is specific for alpha 1, 4 glucose. Very similar to what? To alpha amylase. Nearly. And that, and this is exo. So what do you think is the difference? And always it starts from the non-reducing end. See here, always the alpha amylases break down the polysaccharide into smaller one. Smaller disaccharides and trisaccharides. Then comes the glucoamylase. Glucoamylase, as we said, it is a disaccharidases. It does not function for polysaccharides. No, it is functional for short chain of oligosaccharides and disaccharides or trisaccharides. While alpha amylase is for longer one. The glucoamylase is heavily surrounded by saccharides. Why? To protect it from digestion. And this is most of the enzymes, as you will see in our digestive system, is heavily surrounded by carbohydrates to protect them from digestion from proteases. Proteases are the most powerful enzymes in our body. If these enzymes is not glycosylated, is not surrounded by carbohydrates, then proteases will digest them. Now, this is the glucoamylase. The second example is sucrase isomaltase. Again, this enzyme has 2 catalytic sites, but they are different. Glucoamylase, practice my own. Glucoamylase, the first one, has 2 catalytic sites with similar function. They only break down alpha 1, 4 glucose, that's it. Both functional group. While in sucrase isomaltase, it has 2 catalytic sites, but they are different. The first catalytic site, A, we call it sucrase maltase. And the second catalytic site is isomaltase maltase. From the name, sucrase, maltase. So it breaks down all sucrose, 100% of the sucrose, with maltase activity. It also breaks down some maltose. Isomaltose, maltase, it breaks down all isomaltase, 100%, with some maltose. Clear? So, sucrase isomaltase complex. It has two catalytic sites, but with different function. The first catalytic site, sucrase maltase. From the name, دائما وابدا الانزيم باخذ اسمه من المواد اللي فعالتها. Sucrase, it means it break down sucrose. Maltase, it break down maltose. Isomaltose, it break down isomaltose, alpha-1,6. And maltose, the maltase break down the maltose. Clear? So 80% of the maltose is broken down by this complex, and only 20% is by glucoamylase. Because what is the maltose? Maltose is made up of what? Two glucose. What type of bond? Alpha-1,4. Glucoamylase break down maltose. Maltase, here and here, more functional group, break down maltose. So many enzymes break down maltose. But see here, sucrase isomaltase complex break down 80% of the maltose, while the glucoamylase break down only 20%. Clear? Trihalase. Trihalase is specific for what? From the name. Trihalase is specific for trihalose. Trihalose is a disaccharide made up of two glucose. To which bond? Like alpha-1,1. Glucose. Is it a reducing or non-reducing sugar? Non-reducing sugar. Because both aromatic carbon atoms are involved in the glycosidic bond. Trihalose is broken down by trihalase, and trihalose is a disaccharide that is present in algae, mushroom, some bacteria, and fungi. So it is not well known to us, trihalose. So trihalase has only one catalytic site, see? While the first two enzymes, more two catalytic sites. This one has only one catalytic site. One catalytic site. Trihalase break down which sugar? Trihalose. What type bond in trihalose? Alpha-1 glucose. That's it. And the fourth digestive enzymes for the carbohydrates is glycosidases, beta-glycosidases complex, which is known as lactase, glucose, ceramidase. See here, from the name, the short name, we can call it also lactase. We call it lactase, other name for this enzyme, because it breaks down the lactose sugar. It is specific for which sugar? The lactose. Which bond? Beta-1,4, galactose. Not beta-1,4, glucose. No, it is beta-1,4, galactose, glucose. This is the lactase, the beta-glycosidases complex. It also breaks down the bond, the glycosidic bond, between glucose and galactose, and the lipid, the ceramide. عمرك سمعتوا بالسيراميد؟ ممتاز دكتور. خدناه السنة الأولى، أنا لا. اللي كان ممتاز، يعني لو قلت لا بتكون ممتازين جدا. مثل الأسلاك والسالفين، عمرهم ممتازين. So ceramide is found in the lipid. درسناه وأخذنا عليه بعض الأمثلة. Sphingolipids, the building block of sphingolipid, which is present in cell membrane. A lipid found in the cell membrane. Normally, The ceramide makes a beta bond with the lactose in general. Who break it? This enzyme. So lactase break down the lactose and the beta bond between lactose also and the ceramide. Where this enzyme's the location? See, saccharase isomaltase and beta-glycosidase in the jejunum, while glucometers in the ink. This is where these enzymes are present. Metabolism of sugars by colonic bacteria. Here.
لا نشوف البنات انا كل الاحترام منهم لو حكاها شاب يعني كل الاحترام والله المدن خلنا شنو 10 بس 5 دقائق يعني خلينا نخففوا هذا اقسم بالله لا شيء خلص تسجيل.
See, some dry starch is high in amylose. Starch and high in amylose means it is very dry. Little fasoli, natural. It's very dry. So once you have these beans, our small intestine can't digest them. So they reach colonic bacteria undigested. Who digests them? Colonic bacteria. So these dry polysaccharides, our small intestine can't digest them. Who digests them? Colonic bacteria. Producing what? Gases, short-chain fatty acids, which is very important for the energy in our body, and lactate. This is why once you are sometimes, many of us, once you are eat this dry beans, you feel uncomfortable with some gases. Why? Because the colonic bacteria that they digest these polysaccharides produce this gas. The last thing, lactose intolerance. هذيك الحالة الأولى حالة طبيعية. This is a disease case. Lactose intolerance. From the name, what does it mean? It means that the body of those patients can't digest lactose. This is why there is a deficiency in which enzyme? Lactase. There is deficiency in lactase. This deficiency could be congenital, means genetic, since birth. There is some defect in the gene that produces lactase. This is why their body can't produce the lactase. Or aqua. Aqua is of two types, primary and secondary. Primary has some genetic factors and starts after the age of 2. The real reason is not real. While the secondary lactase is due to infection, or injury to the small intestine, due to injury or infection to the small intestine. So, what is the lactose intolerance? It is a disease case. The body of those patients can't produce enough lactase. He produces some, but not enough to digest all lactose. It could be congenital or acquired. Thus, the lactose reaches the colonic bacteria undigested. Who digests them? Colonic bacteria. Producing what? Methane gas, H2 gas, and lactic acid. This causes what? Abdominal pain, gases, and diarrhea. See? The difference, this is a disease case, not like the first case. So producing abdominal pain, gases, and diarrhea. Clear? How to diagnose those patients? We give the patients a glass of milk or lactose drink. Normally, our breath has very little of the gas, hydrogen gas. We give those patients lactose drink. Then we test the hydrogen in his breath. If it increases, it could be. Second thing, the stool is acidic. The stool is acidic, lactic acid, and some fatty acid also is present. Management, how to manage? Most of this patient produces some lactase. So you know your limit and stick with it. Yani, there are some patients, their body can digest up to quarter glass of milk. Don't do this. If he drink half cup of milk, then he will get those things, diarrhea, gases, and so on. So know your limit, stick to it, and avoid the food that has lactose. Okay. السلام عليكم. إذا عندكم أي سؤال اسألوا تفضل. الله يعافيك، العافية عطوفتك إن شاء الله.